And welcome to the second day of our program. And today session will continues. The question will continue yesterday's talk and the stress mechanisms in the trees. Okay, over to you, Asha.
Okay, thank you, Vishnu.
Yeah.
And
Yeah.
welcome back to second day of our lecture on stress mechanism, how the plants are getting erupted. As we discussed yesterday, so we know basic understanding of physiological mechanism, how the biochemical mechanism will work out, and how the both physiological and biochemical together work into the alteration in the proteins, and that is the building blocks of whole DNA and genetic components. So how ultimately they are getting attached to the genetic level adaptation mechanism. Okay, yesterday, whatever we saw the theoretical part, now I'm going to discuss some of the results which which was a part of my PhD work. So where we wanted to understand how the teak seedlings they are responding or they're behaving to the drought condition or water stress conditions. So here the whole story will be dealt with the two objectives. First one is to understand how the teak accessions. Accession is nothing but the genotypes, how they are responding, the different genotypes, how they are responding to the drought condition. And second part will be like a single nucleotide polymorphism discovery, where we are identifying some of the genes which are really responsible for the drought tolerance and susceptibility in the tea. So the first objective to achieve the first objective, we had 10 different genotypes. They all originated from Kerala and the Tamil Nadu part, and they all replicated statistically everything we conferred. And the ramets, pure true to type ramets produced from 10 different genotypes were used for understanding the stress physiology, biochemistry, as well as the morphological understanding. So this is the experimental setup. Everything was watered to the field level, means the moisture content was maintained, that was recorded regularly, and that for particular time period is considered as a normal period where we have irrigated all the plants to the field level without making them to thrive to the any of the water limited condition. So after maintaining this normal period for the particular of 7 to 10 days that is standardized earlier to the study. So 10 days we maintained plants without irrigating and the soil moisture content was reduced to the 8 to 10%. So since it is a red sandy loamy soil, so we maintain 8 to 10% of the soil moisture content that is considered as a maximum stress period. So, guys, here we are having a normal. Now, after withholding the watering, without watering the plants, we maintain for 5 to 7 days. That was considered as a maximum stress period. Once after all the plants started showing the wilting signs, so that period is considered as a regain period. If you remember yesterday's class, one of the avoidance and tolerance mechanism is resprouting. When water availability is very less or when there is a stress condition, the plants will start to reduce the foliage. So that is the basic concept or the principle we followed here. When the normal condition is there, they will be happily enjoy whole and soul environmental condition. But when there is a limited moisture content, they will show some of the symptoms. The physical morphological symptom is nothing but reduction in the foliage. So that is considered as a maximum stress period. And after the resprout ability, which accession or the genotypes are really, really tolerant or resistant to the drought condition may have a capability to regain after watering to the field condition. So this is the basic common understanding. This is why we are having three phases. One is the normal, one is the stress condition, that is the maximum stress condition, and third one is the regain condition. Here, all the 10 accession you can see in the image, and similarly, the moisture content. of the accession with the whole bag and soil media was checked every morning, and it was maintained for the maximum stress. So same thing was maintained, and the observations, that is a morphological observation, physiological and biochemical observation. First one is the as a morphological observation, only the number of the leaves when maximum stress is there, how much percentage of the leaves were retained. If the plant is really susceptible, then all the foliage will be shut off. If they're having the resistance to the stress, then it can maintain the foliage to achieve the photosynthesis and food production as well as the transpiration. So that's why we have taken the number of the leaf as a morphological parameter. Coming to the photosystem 2, that is PS2, we are having the chlorophyll fluorescence that will tell the efficient of photosystem 2 for making the cells viable for the photosynthesis. So after that, we are having the chlorophyll content and chlorophyll stability, because chlorophyll is major component of the leaf cell uh, for capturing the light, and we are having the photosynthetic rate, stomatal conductance, transpiration rate, and leaf temperature. When there is less availability of water, the leaf temperature will rise, and once the transpiration is getting reduced, the cooling effect of the leaf will be also get affected. So we are having another component, that is canopy air temperature difference. That is a simple difference between the temperature of the leaf to the temperature of the ambient, or the ambient temperature, or the climatic environment temperature. Then we are having the membrane stability. Why membrane stability means when there is a abruption in the biochemical, basically of the lipids, they may get dysfunctional, and membrane may become very fluid or fragile. So that's why we have gone for checking the membrane stability. So these are the morphological and physiological parameters which were evaluated during the normal, maximum stress, and in the regain condition. So this is the infrared gas analyzer where we are checking the photosystems and transpiration rate and temperature. And this is the instrument called chlorophyll fluorometer, which is telling photo efficiency, efficiency of the leaf. Where we, if you are understanding the dark reaction, where we are creating the darkness to the particular spot, and immediately we are passing the infrared light. So if the plant is susceptible and the PS2 is getting damaged, that will recorded here in the fluorometer, and that range will be maintained. Further, we are having the biochemical parameters. They are considered as a osmoregulants, which are important for maintaining the turgidity of the plant cell. So when there is a no moisture, or when there is no availability of water, then we need some of the osmoregulatant for maintaining the uh, ಹೋಗ್ತೀರಾ? Okay, okay. For maintaining the turgidity in the cell. ನಿಮ್ಮ chamber ಅಲ್ಲಿ ಬರ್ತೀನಿ ನಾನು. Okay, okay. Turgidity in the cell, so that the cell will maintain in functional way. So if the plant is not capable of retaining the tolerance, or if it is not resistant to the any of the stress condition, then they may fail to produce these osmoregulatant. For example, basically on one of the important osmoregulatant is proline. Then we are having the glycine betaine. Then we are having MDA, total soluble sugars, and protein, peroxidase, SOD, and catalase. These peroxidase means POD, SOD, and the catalase, they are very, very important for scavenging the ROS. ROS is nothing but yesterday we discussed about the reactive oxygen species, which are considered as a toxic product, which is produced during the high temperature, where the PS2 is getting disassembled and damaged. It will produce the D1 degradation will happen. During that time, there will be production of ROS. These osmoregulants are very, very important for scavenging the ROS. Okay. So these are the glimpse how this will work. So here you can understand, sorry, here you can see how the plants are getting reacted to the drought condition. So this A1, A2, A3, A4 till A10 are the different genotypes which were evaluated for the drought condition. Here you can see the above picture is showing the plants are enjoying maximum water condition. Means we are having the normal condition where they were irrigated fully to the field level, and the below picture is showing how the plants after 10 days of maximum stress. So we withheld the water, and after that we went on for checking the all the parameter whatever we discussed so far. Here in by the view we can see the plant A1 how it is looking, even though the foliage percentage has been reduced, still the plant is capable of maintaining healthiness so that it can go for the transpiration and the photosynthesis. That is the main function of the plants for the food production. So A1 is looking healthy. When further we can see A2 and similarly A10. Here some of the individuals like A8, 6 and 3, the whole foliage has been dried, and A4 especially we can see here no foliage or 0% of foliage has been retained. Only the shoots were getting retained. Maybe it is getting ready for the re-sprouting of the plants after irrigating. So this is the two different stages which is shown as the normal stage as well as the maximum stress. Okay, Melvin, are you guys are following?
Yes,
Yes.
sir.
Yes, sir.
Okay, okay, thank you. So now the thing is that I will not go into the data because that is what is the lowest, highest, and only some of the few things I will discuss here. So, for example, you can see here where in the beginning, in the normal condition, this is the replicated data is pooled, and then we went on the analysis. All the things are followed by completely randomized, blah blah things statistically. Okay, here in the normal condition, we can see the number of the leaves present in the normal condition during the drought and regain phase. Those components were used for calculating the percentage leaf retained. And here we can see some of the individuals or the accessions or the genotypes are not capable of maintaining even 1% of the sorry, below 10% of the foliage. Here you can see the A8, if you are remembering the picture. So here you can see the A8 one, and this is the A4, A2, and A1. Here A1 and A2 are maintaining more than 70%. A3 is maintaining more than 6%. And we are having the uh, categorization that is followed for cocoa. Basically, it's from Cocoa Research Station that is published by Julie and his co-worker by in 2021. Same categorization has been followed here to grouping the individuals into the four classes. One is that is highly tolerant, then moderately tolerant, highly susceptible, and susceptible. So here, whatever the individuals which have maintained foliage more than 70% are considered as a highly tolerant, and more than 45% foliage that is considered as a moderately tolerant, and less than 30 to 25% that is susceptible, and less than 25% is considered as a highly susceptible. So only one individual out of the 10 is highly susceptible. This is only based on the morphological feature. So we have considered only the number of the leaves. So for the preliminary confirmation. So here we are having different components like photosynthetic rate, stomatal conductance, transpiration. I will not go into the data, but I will tell one or two facts that even though in the normal to drought condition, there is a slight variation in the respiration or the stomatal activity and the photosynthesis, but still some of the individuals are capable of maintaining high photosynthetic rate compared to some individuals. That is the only one thing. Means even though there is less water available for the plant is capable of producing some other components in absence of water molecules, and they are maintaining the photosystem. So this is the basic thing. So here what happened, basically A1, A2, and A7 you can see under the drought condition also it is maintaining more or less normal condition of the photosynthesis. means the drought has been not impacted the photosystem activity. Similarly, for the stomatal conductance and transpiration, even though there is no availability of moisture, plant is making the transpiration, evapotranspiration will be there, and transpiration will be also there from the plant foliage. Similarly, for the chlorophyll fluorescence, that is telling about the photosystem 2 and water content availability. See, even though there is no available of moisture in the environment or the soil media, the plant cells are capable of maintaining some of the maintaining the moisture content. That is because of the osmoregulating what we discussed, that is the proline, glycine, betaine, and MDA basically. And here fluorescence also, the activity of the PS2 is normal. It is compared to the normal and drought condition, it is more or less similar only. So the chlorophyll activity and chlorophyll fluorescence has been not influenced by the drought condition because they are might be susceptible, tolerant. Some of the individuals may be susceptible. Here the biochemical parameters, that is the proline, glycine, and MDA. You can see the all the osmoregulants has been increased from normal to drought condition. That is one observation. Means during the normal condition, when there is a plenty of the water molecule available or water availability is there, there is no need of production of any of the osmoregulatant, because for the production of osmoregulatant, again plant need to consume some energy or waste some energy. Instead of wasting energy for production of these osmoregulatant, they will say go for the plant development and growth. But when there is a drought condition or water limited condition, then they will think of maintaining the fertility and healthiness of the plant so that it can survive. So here you can see under the drought condition, the proline content has been increased. The maximum has been found in A1 and A2, and similarly glycine betaine and MDA formation, everything has been found increased in some of the accessions, and comparatively the A8 accession, the eighth accession is reduced very minimal osmoregulatant. And here you can see the grouping. So the dendrogram, here it after understanding only under the drought period, it is the dendrogram was constructed only considering the drought period. Here you can see the accession 8 was classified separately, and A1, A37 to 1095, they all some of the individuals which shown tolerance, which shown high percentage of the foliage, and which are shown for the increased production of the osmoregulants. So they classified into the different classes. So for proving this one statistically, we have gone for the repeated PCA, that is nothing but principal component analysis. Here, the method we followed for categorizing the individuals. See, first one we categorized based on the morphological itself, based on the number of the leaves. Now here again we combined all the morphophysiological and biochemical parameters, and we went on the PCA, and those PCA scores again repeatedly we go for repeated principal component analysis for giving the index score. Index score is nothing but here it's again by considering the PC values and every loading values we constructed the importance of each parameter, and the weightage has been given, and those scores were formulated and final scores are depicted here. And this is the grouping of the individuals based on the scoring level. So here this is a PCA by plot. You can see the eight accession which were not capable of maintaining the foliage or which could not produce any of the osmoregulatant has been grouped separately. The same grouping has found in the dendrogram, and dendrogram is constructed differently and PCA is constructed differently. For confirming the groups only, we have gone for the different kind of the statistical analysis to come to the final decision. So here 6 and 4 are considered as a susceptible, and A8 is considered as a highly susceptible with the index score of 44, and susceptibles were had the score of 62 to 68. Then we are having most of the groups or most of the individual, the four individuals are falling in a same category of moderately tolerant. That is accession 9 with a score 82, accession 7 75, 391 and 93. Only the accession above 95 were considered as a highly tolerant, that is 2, 5, and 10. The 2 was considered as highly tolerant with the A5 106 and one more 97.76, that is the A10. So here the thing is that some of the if we are considering only two classes, susceptible and tolerance, whatever the half of the biplot individuals are there, they're all considered as a tolerant, and the whatever the left side one is there, they will be considered as a susceptible if considering into the two classes. But we made it into the four classes, which will helping us for the genetic identification of the individuals and identifying the genes. Is this clear so far? Whatever we discussed, whatever we told yesterday, physiological, morphological, and biochemical, we have studied those parameters and we came to the conclusion. Out of the 10, we categorized them into the susceptible and tolerant classes. Yes, Melvin? Hello? Adira?
Yes, sir, it's clear.
Okay. Are you guys listening or?
Yes, sir.
Okay.
Yes, sir.
Okay, here we completed a half story where we wanted to identify some of the tolerant and susceptible. Now we have to go for understanding the genetic level, why these individuals are really tolerant for the drought condition and why some are susceptible. So here we adopted the principle called GWAS, that is genome-wide association studies, where we identified single nucleotide polymorphism. So you aware about the nucleotides ATGC. So they are the
Yes, sir.
building blocks of our DNA. If see most more or less 99% of the components or the composition will be similar in the whole human being. Only 1% may make vary, which leads to skin color or hair or whatever the thing. So that particular thing is called as SNP. Are you guys heard about the SNP, single nucleotide polymorphism? Anyone working on the genetics? Okay, no problem. Single nucleotide polymorphism means, for example, if you are taking, if I'm taking 10 nucleotide that is having A, T, T, C, G, C, if there is any one change in the one nucleotide, that will make change in the whole, that will cause the variation in the whole organism. So that variation, that is called as a polymorphism at a single nucleotide. So that's why it is called as a single nucleotide polymorphism. And G was nothing but genome-wide association studies, nothing but throughout the genome we are identifying where exactly it is having the difference, or where exactly that single nucleotide has been changed or that variation has been brought. Okay, here we are having three stages. One is the phenotyping, whatever the story I spoke so far, the biochemical parameters and physiological, and coming to the classification of the individual. So drought classes we made, whatever the tolerant, susceptible, and everything what we're talking. So that is about the tolerant classes. And this part is called as phenotyping. And we are having the process called genotyping, means we have to get the gene sequence. So what we are doing, we are extracting the DNA, then we are going for the cDNA library preparation, then Illumina Nova sequence, that is a technique for the sequencing the genotypes. After sequencing, the genotypic data will be ready. When the phenotypic data and genotypic data are ready, then we will go for the genome-wide association study, where the 10 genomes, whatever we extracted and we went on the sequencing, were compared with the reference genome available in the NCBR or Giga database. That is, recently, whole genome of the T has been sequenced and that is available in the library. So that is considered as a draft genome. When how the T genome, the reference genome has been constructed, that is having the n number of individuals. It is not considered by considering a single plant. It is considered more or less 1,000 plants, and then they will go for constructing a reference genome that will be like a reference for the whatever genomic work we are going to do. And with that reference genome, we compared our sequence data of 10 genomes. So then that process is totally something like here variant calling. Variant is nothing but where it is exactly having the variation. That's all. Don't worry about that one. Finally, what we will get there, that is the location where exactly the nucleotide has been changed. Okay. Here you can see this is the 19 chromosomes. Even though the teak is having 18 chromosomes, we are having a pseudochromosome that is considered as a 19. So these are the locations of 10 genotypes where they have observed with the different nucleotides. And after getting the nucleotides, because see, we identified more than 3,000 nucleotides which are totally different from the draft genome, but all the 3,000 will be not responsible for the adaptation mechanism in the T. So what we have to do, we have gone for the generalized linear model we used, and we proved that one through QQ plot, that is quartile plot, and then we the result was same result we expected from the rectangular Manhattan plot. Here, based on the logarithm value, that is the e value of the each individual SNP, we have identified some 10 to 15 or 20 SNPs. They are really linked with the drought tolerant classes. So these genotypic values and our drought tolerance value, whatever the four classes are there, they were combined, and those SNPs which were considered with the tolerant lines only picked up here. These are the individual SNPs which are found highly suitable for the tolerance of the individual. So after getting this particular SNP, now we know which location is this SNP is having. So this is the location shown here. Okay. So this is the chromosome number one. We are having 171 to 268, but they are all not exactly SNPs which are responsible for the drought tolerance. But these SNPs finally, after GLM score SNP, whatever we got, they are the responsible for the whatever the physiological or biochemical changes happened in the tolerant classes. So here you can see this is the QTS with the given chromosome number, and this is the position. So including the chromosome number and the position that the QTL name has been given. So these are the nucleotides found in the reference genome of the teak, but these are the nucleotides which were found in the genotypes which were drought tolerant. You got the point now? So this is the reference, and this is the nucleotide which found in the drought tolerant genotypes. So after coming to know, after letting know of the alternative nucleotides here, then we went on for going the annotation of the individual QTS. So we came to know we are having some aberrations at the genetic level. If that change is there, what exactly that change will bring in the genetic level? So that we wanted to know. We went for the blasting the particular QTL. That is again the genotypic work. And then after getting the genes, during the blast, we will come to know what exactly genes are. We will get to know the name of the genes. After getting the name of the genes, then we have to go for identifying the functions. That is called as a functional annotation that is done in the Uniport database. So here I highlighted some of the QTLs and the genes. Even though these genes are responsible and found tightly linked with the drought tolerance, we could not come to a conclusion that they're having a playing a role in the drought tolerance. But whatever the things I highlighted here, for example, chromosome number 4, that is 2520146, is responsible for the production of the genes called deoxyxylose 5-phosphate synthase, and this gene is having a role in the chlorophyll biosynthesis. Now we go to the point why the chlorophyll is important. Again, it is for capturing the light molecules. If the tolerant are capable of maintaining D5 pH, and that will help in the production of the chlorophyll during the drought period. Even the foliage is less, the minimal foliage will be maintaining the chlorophyll biosynthesis, and that will produce the food for maintaining the healthy or the resistance to the plant. Similarly, we are having the chloroplast and chloroplast stroma, that is again for capturing the light. Here in the chromosome number 1, we are having bell-like homeodomain protein 9, that is BHP9. We are having what is the role of the BHP9, that is responsible for the plant organ and secondary shoot formation, the mechanism for the resprouting. So after the drought period, when we regain the individuals, some of the individuals will come back to the normal period. So that is why this BHP9 is very, very important in the tolerant classes. So they are maintaining the plant organ and secondary shoot formation, xylem development, and morphosis. Morphosis is nothing but resprouting or the maintaining the morphological things. Then we are having the POX, that is plant homeobox family protein, that is very, very important for the organ abscission and meristem maintenance. The organ abscission is nothing but falling up of the leaves is one of the first and foremost symptom in the drought condition. So when the POX family protein has been produced during the drought period, immediately plants will get a signal. So we have to go for the organ abscission. We have to shed off the leaves to maintain the moisture content or maintaining the healthiness in the plants. So organ abscission is the function of the POX family protein. Then we are having the major facilitator superfamily domain containing, that is important for stomatal closure, and probellucin rich repeat receptor like protein kinase. They are important for the root activity. Root activity in the sense when it is in a field level, some of the individuals can thrive into the deeper layer of the moist soil, deeper layer of the soil profile, and they can absorb the moisture, or the root hairs may be having some mechanism to drop out the drought condition. That's why root cellular catalytic process is maintained in the chromosome number 11. And then we are having MDIS1 interacting receptor, that is important for the cellular and defense response for insect and hormone mediated signaling pathway. Because whether it is an insect, biotic or abiotic, that the signal pathway will be important for maintaining the healthiness. So this is the ultimate thing we found in the tolerant classes. We are having such genes. That's why these individuals are becoming the important tolerance, or they're becoming tolerant to the drought condition. So this is the in nutshell. Almost I reached the end of the presentation now. Based on the selection index, that is in morphophysiological and biochemical parameter, we found some of the individuals are tolerant, susceptible, and moderately tolerant and moderately susceptible. This is the okay. Leave it the thing. The QTL, which found for the BLP, BHP9, and DXS and MFS family major facilitator superfamily. They are the important thing. So ultimately, thing is that now after understanding these important genes are causing the drought tolerance in the teak. So what is the next stage we have to do? See, maybe the teak which are growing better may not be ready for the upcoming stress condition. So what we have to do, in the simple way I am telling, you might have seen the Neelambu teak. here in our study, the teak from Kerala, that is from Neelambur, found very, very susceptible because it is maintained, it is got in throughout the evolutionary process, it has been not undergone for any of the drought condition. So it is enjoying its natural habitat and that got adapted to that particular condition. But the quality of the teak produced is number one, but if that plant want to be grown in the Rajasthan, that is not capable of withstanding the heat or the water scarcity there. Now, what we have to do, we have to go for the genetic modification through incorporating these minimum genes to that particular plant so that we can achieve better growth performing with having the drought tolerance planting material. So this is the only one thing we can do here. Otherwise, if we are most of the time, what will happen? Genetic modification is totally against, and there is a pro, and always there is a debates going on genetic modification GMOs. We identified some of the individuals which are tolerant. Those individuals can be propagated through tissue culture or vegetative. Then it can be planted in the sites where it is having a moisture stress or the stress condition. So now I will reveal these things, that is the accession number. Here you can see that A2, A10, and maybe A4. These are the some of the individuals, sorry, A3. They are some of the individuals which show the tolerance to the drought condition. But the thing is that, guys, that is evolved and selected from the Tamil Nadu region because the plant grown there with the water scarcity has undergone to the evolution and they change the genetic makeup and they are shown the tolerance. A1 was moderately tolerant, A2 was highly tolerant. Then we are having AA from the Nelikuta or Nedumkayam area, that is from the Neelambur area. They found highly susceptible or susceptible for the drought condition. So this is the final conclusion of our study. So understanding how we can understand the mechanism of adaptation and evolution throughout the things. Okay, thank you everyone for the patient listening. So thanks Igor for giving a chance to deliver my lecture series. And I also thank the Department of Science and Technology. I was a DST Inspire Fellow for my PhD work and that got DST Fellowship, DST Inspire, and funded by NFST and Kerala Agriculture University, KFRI, and IFGTB and our own university. End of the class of the session. If you are having any of the doubts, you can drop here, or if you want to understand something else, you can put it in a chat, or you can unmute yourself and you can talk.
Hello, Ashwin. Can you hear me, Dr.
Yes,
Ashwin?
yes, I can hear you. Yes.
Okay. So, Dr. Ashwin, I think there are few.
Yeah, someone put that Arundhati put some message.
Yeah.
I have common doubt. That is a common doubt, and that is proven. Even if you are reading some of the, I forgot the book, name of the book, where they compared a mango tree. They compared a mango tree which was not producing fruits, which were not producing fruit, and he went on to talking to the plants, and he was talking very nicely to one plant, and he was shouting at another plant, and whatever the plant he was talking nicely, and it started to flower, and this is proven actually. And now people are understanding the language of the plants through the electric signals. Hello. Arundhati, we can discuss, or I think I can share some of the material. If you're putting a WhatsApp me, we will
Okay,
share it
okay,
and we
sir.
can discuss. Yes.
Okay, thank you, sir.
Okay, thank you. Thank you.
Okay, thank you, Arundhati, for the question. And is any other question from the participants? Anyone else? Hello. Any other questions? Okay, Ashant, I think there is no more questions from the student side. And I hope our students can reach out to you if they have any questions in the future related to the plant
Okay.
science. Isn't it?
Yes, yes, yes.
Okay,
I
then.
think they can give some feedback. Maybe to you, you can forward to me
Okay, definitely.
because that
I
is
will
very
do.
important for us also.
Yes, definitely. And there is one more questions from
Hello,
Ruxana.
sir. Where the SNPs
SNPS expressed
exist?
similarly in all other drought tolerant plants. Yeah,
No,
please
actually,
continue.
see, here again, the thing is that when the tolerant classes were there, we are going to score it. I didn't touch most of the genotypic things there. When we scored them with the base of 10.5 with the classification. So only the tolerant classes, that is not a mean value. Whatever the SNP is found among the four tolerant classes, whatever the things, that's why I got around 20 positively linked SNPs. But what they exactly working, that one only I highlighted, maybe 4 to 5. That is only what we needed. Maybe other SNPs had other roles to play. Some shown for the biotic stress, that is not very important, so that is not from one plant, or it is not from all the plants that we scored and we grouped them into the different classes. Then we went for understanding the SNPs.
Okay, sir.
Yes. Uh, if there is no more questions, I think we can wind the session.
Yes, Session, I think there is no further questions from our students yet. And maybe they can reach out to you through your mobile number or the Gmail ID. I would like to express my sincere gratitude and to you for accepting the invitations in your busy schedule and giving two sessions related to the plan and its stress. And I think it was your PhD program's topic, and you could share your topic with our students. And thank you so much. So we are expecting your valuable sessions in the futures as well. Okay, and
Yeah.
hopefully,
Thank you.
hopefully, yeah,
Yes, yes, yes.
hopefully we will conduct one maybe a hand-on training in Karnataka at some point of time in your free time, as we discussed earlier.
Okay, okay, okay,
Okay,
okay.
okay
That's
then.
fine.
Yeah.
So
Thank you.
basically, the place where I'm staying is nearby Hampi. You might be knowing all the guys. If you're
Yeah.
reaching out here to the Hampi, you can contact me. We can meet here also. Anyone,
Yeah.
interns.
Yeah. Interns, they can mostly we will conduct some training programs in different part of India. If you're conducting in your place, then definitely we will come to Hampi
Okay.
and stay there. Okay.
Yes, yes,
Okay.
yes. Thank
Okay.
you, and thank you to the
Yeah.
Eco,
Thank.
and thank you to you also, Jishnu. Thank you
Yeah.
for the approach. Okay, then.
Yeah. Okay.
Bye.
Thank you. Thank you so much. Thank you. Thank you then. So we will wind up our today's session, and the interns have the next sessions in the next week, and that will be related to the environmental toxicology, ecotoxicology, and its biology. So it will be by Mr. Vasantakumaran. He is doing his PhD in marine biology. Okay. See you next week. Okay. Thank you. Thank you then.